Hello, everyone. I would like to talk and remind us why we like DNA, because my preferred title is Why We Love DNA. Because we often ask, or I'm often asked, why do you use DNA? There are so many molecules, so many polymers. Why do we use DNA as a digital tool? And I'm aware you all know this, but I think we have to remind ourselves every now and then what are these unique properties, chemical properties that this molecule brings with it. The most important is Watson-Crick base pairing. That G binds to C and A to T is a key chemical property that we use in many underlying tools or technologies, and I'll quickly go through them. One is toe-hold replacement. I won't talk about that, but that's, let's say, key invention based on Watson-Crick base pairing, used a lot in DNA computing. But we also use this Watson-Crick base pairing in many, many other of the tools that we are using. As said in the introduction, I'm a chemist. And as a chemist, we, let's say, often in analytical chemistry, or always in analytical chemistry, we combine chemical information and, let's say, digital information. We put a molecule in, and then we want to know something about the molecule. And if we want to use chemistry as a digital tool, we kind of have to ask the other way around is usually what we do. Our question is, how do we get the most information out of a molecule? It's not that we have a molecule and we want to understand it, but we want to get information in and out of molecules. And as a chemist, if we look at analytical properties and molecules that we can look at, DNA is really unique, because DNA is the only molecule where we can detect one single molecule. And this is often forgotten. If we, for example, if we want to detect gold with mass spec or so, we need enormous infrastructure machines. Okay, thanks. If you want to measure gold, we need enormous machines, and we need thousands of gold atoms at the same time to be able to say that's gold. With DNA, we can detect only one molecule by PCR, and we can not only detect it, we can read the whole information, let's say the sequence, by sequencing technologies. So we get about 200 bits of information of 0.1 attogram of mass. So we are really good at measuring this molecule. And a second property that doesn't rely on Watson-Crick base pairing, but is also unique to DNA, is that we are really good at synthesizing it. As you know, usually when we synthesize DNA, it's like playing Lego. We put one block on top of each other. Those involved in the chemistry of it know it's much more complicated. Let's say we have these circular reactions where we take these building blocks and we place them one on top of each other to make the molecule that we are interested in. But where this is also possible with peptides and other polymers, what's really unique to DNA that has been parallelized, and we can synthesize thousands or millions of different oligos, DNA molecules of different sequences at the same time with different miniaturization technologies such as printing, electrochemical, or light-directed DNA synthesis. And this is not, there's no other molecule where this is possible at this scale as we have with DNA. So this doesn't depend on Watson-Crick base pairing, but it's really unique to DNA as a digital tool. Also, and that's really based on Watson-Crick base pairing, where we know Watson-Crick base pairing best, is PCR amplification. Where in PCR amplification, we have a DNA molecule and we can make copies of it. We take primers, they bind to the, let's say, sequence of interest, and then we can amplify the DNA molecule exponentially. We can do that in two different ways. Either we have a fairly large pool which has common priming sequences at the end, as shown here at the end of this paper by Nick Goldman, who, let's say, restarted this field of DNA data storage. Or we can use primers which are somewhere within a sequence, for example, in a phase-specific part of the sequence and only amplify one individual sequence out of a large pool. Or we can play games in between, and I'll get back to that. And we all, or many of us certainly rely on PCR as a tool to amplify molecules in many of the things we do. And then we have to ask ourselves, do all sequences amplify to the same extent? And there's been some work also in the field of DNA data storage that showed that that's not really true. So this is an example of the paper of Janif Ehrlich, where he had a pool of DNA sequences, all with constant primers, and he amplified them not once, not twice, but I think 10 times or so, 15 cycles every time, and then looked at the composition of the sequences after that copying processes. And the distribution of sequences is completely skewed, meaning that some sequences amplify better than others, which is against our, let's say, belief that we think that PCR is this perfect technology that just amplifies all sequences, like we take electrical signal and we can amplify it to some extent. We also see that in some other DNA data storage works, not always to such an extreme extent as in the work of Janif. Depends a little on what, let's say, primers, polymerases and so you use, but it is a common problem we observe in many of our workflows and also in biological workflows. So we were interested in understanding why do these amplify differently? What is the reason behind that? And to be able to do that, we generated large pools of DNA sequences, as depicted here, with all the same constant primer at the end. They have a random insert, so there's some, let's say, sequence information there. They're constant primers, and we then do cycles of PCR in a phase-controlled way, and we measure the distribution of the sequences after every of those PCR runs. And we then, by measuring the abundancy of a specific sequence over that series of sequencing runs, we can estimate the PCR efficiency of every sequence in the pool. So those who've done PCR in the lab, we usually do that with single sequences by, let's say, dilution curve. Here we do that exercise with a large pool, 10,000 of sequences at the same time. We measure the efficiency of every individual sequence. And we want to correlate that to the information that's in the sequence. So how does the efficiency correlate to the information? And to solve that, we collaborated with a group in ETH and now Max Planck Institute, Carsten Borgward, who's an expert in machine learning, because that's a problem that seems suitable for machine learning. We want to correlate an output to a lot of information. They used a 1D CNN model to correlate our, let's say, sequencing, our PCR efficiency to the information of the sequence. And we can see from this data that we get a relatively good performance in predicting PCR efficiency from sequence information. Those who've done machine learning know this is a black box at the onset. We don't know why, what are the properties behind it. But this group also did quite some work in investigating what motifs are then responsible. So understanding this black box of the machine learning, what motifs are responsible for the preferential amplification of some sequences over others. And then there's a motif discovery. We see that the specific motifs, when they are present in the DNA sequences, the sequence doesn't amplify very well. And also we found out that these sequences are usually at the beginning and at the end of the sequence, meaning there might be some correlation with the primer sequence that is responsible for where the primer binds and amplifying. You see that here, we really see these sequences of a specific motif really appearing at the beginning or at the end of the sequence. And we then can correlate that to biological or biochemical mechanism, where although our sequences are which prevent amplification are extremely short, they're only 4 or 5 bases long, they can still impede amplification, which happens at really high temperatures, where you would expect that these short interactions of Watson-Crick base pairing are not relevant at those high temperatures. But we can really see that sequences which at the beginning or end are similar to the amplification sites, they are impeded in their amplification efficiency. This also goes then back, depends on the primers, on the enzymes used, but we have, let's say, really shown that this is a mechanism that results in the impedance of amplification. And we can show that we could, if we can remove those sequences from the pool, we can, let's say, have a much more homogeneous distribution of sequences in the pool, and the amount of sequencing we have to do to observe, let's say, 99% of the sequence is drastically removed if we remove those sequences. I know in, let's say, DNA coding, people try to prevent such problems, and usually people try to avoid GC imbalance or polynucleotide repeats. However, we can show here that these, let's say, motifs that we have identified, they're much more important for this, let's say, for the whole workflow than an imbalance in GC or in polynucleotide repeats. And we've, let's say, have this workflow which we can now apply to many, this machine learning workflow and experimental workflow we can apply to many, to understanding better many of the problems we have in the pipeline, starting from digital information, going to DNA, back to digital information, and understanding really the insights into the problems that are in this in those processes. So I talked about PCR amplification, synthesis, sequencing. So we really have this molecule which has these amazing properties, mainly based on Watson-Crick base pairing. However, also as a chemist, a big problem we have with DNA, or I see with DNA as a tool, as a chemical tool, is its stability. Here we kind of have a chimeric situation, because we expect that DNA is really stable. We have, let's say, these million-year-old fossils where DNA has been recovered and whole genomes of mammoths have been recovered from those samples. On the other side, we know in the lab, if we work with DNA, DNA is not really stable. If we're not careful, so instructions for handling DNA is to put it in the freezer all of the time. Otherwise, it decays. We also know that in our body, DNA is in constant decay, constantly errors happening to the DNA, and we have enzymes which repair that DNA, ensuring that we live a long life without cancer. And so we want to understand what is this, why do we have these, let's say, two different worlds, and how could we go and connect them? And for that, some chemistry, so we have to understand what happens if DNA decays. So if we have a DNA strand, the most important chemical reaction that happens is reaction with water. And if the DNA reacts with water, we lose a base, and there's an A basic site, and these A basic sites are not really stable. The DNA then breaks at the site of this A basic site very quickly. So this is a relatively slow process. The second process is really fast. And so the DNA breaks into two parts. And the important message in that also is that this depends greatly on the length of DNA, because the rate is per base. If you have 100 bases, it's going to happen at the rate 100 times of that of a single base. If you have 1,000 base long DNA or 10,000 base long DNA, it's going to happen much faster that your DNA breaks into two pieces. The rate per position is the same, but that you break your DNA into two pieces, each 10 times faster for 1,000 mer than for 100 mer. And we can really relate that one-to-one to the rate of an individual site. This has also been investigated quite well in, let's say, ancient DNA world, and we look at the lengths people look at in ancient DNA. So this is for a sample, that was 2 million years at minus 17 degrees, some bone sample. The DNA that's recovered is perhaps 40, 50 nucleotides long. Starting from genomic DNA of that animal, it breaks down, and what in the end is analyzed are these really short pieces. And if we look at that at room temperature, we measured that, so that's DNA 150 mer, at room temperature is decays. So that's the room temperature, that's freezer data. So after a year, 90% of the DNA of 150 mer is broken. So we really have to address this problem of looking at DNA, and there's going to be a session afterwards and really nice data that's going to be presented on much, much longer data than I have here on how we can preserve DNA by preventing its reaction with water. Because we want to prevent things at home or in our kitchen. These are usually the things we do or the technologies we have access to, and with DNA we use very similar technologies. So we can either put the DNA into the freezer, we can put the DNA into capsules and remove the water and oxygen from the capsule so the DNA has nothing to react with, or what, let's say, I've been working on is that we put the DNA into something solid and prevent decay as very similar to the situation that DNA has in bones, where it's protected by the bone from decay. Also, my picture is famously leading is never in amber. Amber is much too old. Amber is like 100 million years old. In Jurassic Park that exists in fiction, but in real life DNA is never in amber. It's always in bone, tooth, really solid, resilient inorganic materials that prevent the decay of DNA. We've shown that we can prevent the decay of DNA with very similar technologies as we find in nature. Calcium phosphate. Calcium phosphate is the inorganic part of our bone. And if we very carefully and very slowly dry DNA together with calcium phosphate, the calcium phosphate forms crystalline structures, as you can show here. And within this crystalline structure, you can even see, let's say, the DNA strand. So this is a genomic DNA, so very long DNA, the decay of the DNA is prevented. But calcium phosphate, for technical applications, is not the most resilient material. It's very sensitive to acid and base. We would like to have something better. So we developed a technology to encapsulating DNA into a really resilient material. And the material we chose to encapsulate DNA is glass, because glass is chemically the material which is the most resilient. As a chemist, our cupboards are full of glass bottles because all chemicals we store, they don't react with the glass bottle. That's why we like glass. It's a really resilient, really inert material. And we want to get DNA into glass. So the technology we invented is we take a small glass nanoparticle, which is positively charged. We add DNA. The DNA, as you all know, is negatively charged. It'll bind to the surface of that small glass particle. We use this chemical, which has a positive head, which will bind to the negative DNA on the surface. And on the other side, it's, let's say, looks a little like silica, like glass, but it's a metallo-organic precursor. And we bind that to the surface. And then we use the second chemical, which looks like glass is SiO2, quartz glass. And we use this decomposes and polymerizes to glass on the surface of the DNA that we have on the surface of our particle. So in the end, what we do, we polymerize glass on top of DNA. So we end up with, let's say, DNA molecule with glass on the inside and glass on the outside, so it's really packed in to very small particles. That's what the particles look like. They're like 150 nanometers in size. You can here see the original particle, a very thin layer, that's where the DNA is. And we have, let's say, because these particles are so small, if you have a milligram of particles, we have 10 to the 14 of these particles, so we can get quite some DNA into these particles. And if we store DNA in this way, in these particles, the DNA is as stable, or we think it's as stable because we have to extrapolate by rapid aging tests, as stable as DNA found in fossil bones. So a few hundred years half-life that we get with DNA in these particles. I think we're going to see later that if you really get rid of water and any, let's say, chemical reactants, you can get even higher stability than we can obtain in these particles. But in this particle, we can get a few hundred years of half-life DNA. And in collaboration with Microsoft, we then use this as a technology to think about how would we store files in DNA. What would we do using these particles? And the technology we looked at with them is taking DNA libraries, putting them into particles, then putting those particles onto a digital microfluidic device and storing, let's say, glass slides fabricated by that digital device. And we can show that on storage on that device, that the stability of the DNA in the particles is as stable as the stability of DNA at minus 80 degrees. So as opposed to at room temperature, where we lose 90% in a year, encapsulated and on a glass surface, this DNA is really stable. And just to show you the microfluidic device, because I think it's always nice, I hope the movie works. No, sorry, let's wait for a second. No, sorry for that. But you see, let's say, the droplets of particles moving around on that digital device. And this is just an image we really like to show the size difference of storing things in DNA as opposed to traditional technologies. So this is these DNA particles, which is, the particle is mostly silica and has a little DNA inside on a DVD surface. And in every of the particles, we have, let's say, about 100 DNA strands, so 10,000 bits of information in each particle. And on a DVD, I'm sure many of you know more about coding in, let's say, how much information is in the track. As far as I could find, one of these holes is around 3 bits of information. So if we sum up on this image, we have about 160 bits of data on the DVD, and we have more than 1 million bits of data stored in DNA, just to show on one image how dense DNA is as a storage technology. Even if we have the DNA encapsulated and protected, it will still decay. I said our half-life is, let's say, 500 years. So after 500 years, half of the DNA will be decayed. There's still some residual moisture or chemical reactivity in those particles that results in decay of the DNA. But I also told you at the beginning that in our body, the DNA is constantly repaired. So the question was, can we use those technologies of DNA repair and combine them with DNA data storage to make DNA, even if it has been, can we repair the decay that has happened? And to show the problem a little more in detail, a big problem we have with DNA, if we store information in DNA, is if the DNA strand breaks. At least how we store DNA, we have an index in the DNA, stored in the DNA, and if the DNA breaks, the index is no longer connected to the information. If you have mutations in DNA data storage, that's not so bad, because mutations, we have error correction codes, they can easily handle those mutations. But breakage is really a problem. A DNA strand that's broken, the whole information that's stored in the DNA is completely lost. So the question is, can we use these enzymes to repair this? Can we take this nicked DNA that's, let's say we lost the base, we have double-stranded DNA, so it's kind of still keeping together as we store it, as we then analyze it, it's, let's say, we denature it and it falls apart, but as long as we store it, it's still in this double-stranded fashion, and we wanted to see if we can repair that. To be able to do that, we have to understand the decay a little better. If we look at the decay, so we have this mixed base, and then the sugar opens, which forms this unsaturated aldehyde. And most enzymes can't work with this unsaturated aldehyde. So it really blocks, because you could think that you just take this, you take a polymerase or ligase, and it just fixes the holes. But polymers and ligases, they can't work with this unsaturated aldehyde. We first have to remove the unsaturated aldehyde with a phase-specific enzyme, AP1. And once that is removed, we can use traditional polymerases and ligases to fix the hole we have in between. We can really fix the nick. And just to show you that that works, this is a DNA library. We then age it, where the DNA library essentially disappears because it all smears out. They're all different lengths. They break. But then if we add the enzymes, we can stitch it together again. And we get back the original lengths of DNA molecules that we had. So this is in a gel. We can also look at that in a sequencing data. So this is the length of oligo pools. Our original pool is the dotted dark blue line. So it has about 22% of proper length oligos. This is not the nicest DNA pool you've ever seen, but that's what we looked at here. If we age it, it completely disappears, the proper length, at least at those aging conditions, a few weeks at 40 degrees. And then with repair, we can get back some of the original length products. We can't get back all of them. But we can, let's say, DNA that would be otherwise completely lost for analysis and sequencing in our case and decoding. We can, let's say, tie the knots together again and get back to a useful DNA pool just by adding the right mix of enzymes to our DNA pool. And so we can reverse some of the decay. So in addition to this, let's say, general knowledge we have on DNA, we have a very good understanding nowadays how it decays, why it decays, how we can prevent decay, and even if it decays, how we can revert some of the decay in, let's say, under specific conditions. So for the next part, I want to talk to you a little, what do we do with all of that? So I've shown you, you know most of that, Watson-Crick base pairing, sequencing, and synthesis, but what can we do with it? What we do with it, I'll talk shortly about DNA data storage. I won't talk about DNA computing because I don't know a lot about DNA computing. I'll talk about things we've done in the past, which is DNA barcoding and DNA of things. And in the end, something I'm really interested in at the moment is DNA cryptography, or what we call chemical unique functions. And I'll show you, try to give you some insights into that at the end. When we do DNA data storage in my lab, we do DNA data storage by base storage. So we encode digital information into a base. We have worked with computer scientists for encoding technologies. We have looked into synthesis. I talked to you about amplification and the biases amplification brings. We haven't looked into sequencing. We do Illumina sequencing. We are, of course, people nowadays do a lot of nanopore sequencing. We really like Illumina because it's really reliable and the error rates are really low. And then again, we work with computer scientists for the decoding. But there's going to be a lot of, let's say, presentations about DNA data storage, so I won't go very deep into this, but I want to talk to you about, let's say, our start into this field, which is DNA barcoding. Because one problem we always see with DNA data storage is a big bottleneck is the cost of synthesis. If you want to store a megabyte, it's expensive. And the question then is, well, if a megabyte is expensive, is a kilobyte also expensive? Is 100 bytes expensive? And so we are really looking into, we have all of these wonderful properties of DNA. Can we make them useful other than storing gigabytes of information? Is there usefulness in combining a little digital information that we can access at low cost and our, let's say, technical world we live in? And the most extreme case into that is that we say we only use a barcode, 100 bits, just one sequence, where we, let's say, put some digital information in those 100 bits, which can just be a barcode number, just something. And we use those barcodes to put it into products. And that's what, as mentioned at the beginning, our spin-off company Helixer is commercializing, is taking DNA in these particles to ensure that the DNA is really stable and can survive the whole life cycle of a product and also the whole manufacturing process of a product and really put a barcode into the product. Because all other barcode technologies we know, they're on the product. If I buy a bar of chocolate, there's a barcode on it. But as soon as I remove the packaging, I don't know anything about the bar of chocolate. with DNA, we can really go into the bar of chocolate and write a barcode inside the chocolate. We don't do that with chocolate, we don't do that with food. There's regulations that have to be overcome to be able to do that in food, but we do that with textiles and other materials. And in textiles materials, stability is extremely, so the DNA stability is extremely important because the chemistry that goes into textile manufacturing is very harsh. So in our case, the DNA is sprayed onto the cotton at the farm or at the, let's say, first site where the cotton is collected, and then it goes through ginning, spinning, bleaching, manufacturing, and in the end we want that the final t-shirt still contains the barcode within the t-shirt. Where does the cotton come from? And we can get, let's say, information on, we can add information at different sites to understand the supply chain of this product. Because what we've learned, many of these large textile companies, they don't know their supply chain themselves. Somebody orders some stuff somewhere, which is chinned somewhere, and in the end they sell it in a store. But these companies want to understand the supply chains better, and for that, this DNA barcoding is a really useful technology. Helix has now worked with a few companies. One is in cashmere, of course a high-value product, but now they're also working with C&A, so really bulk product, to enable that the value added by fair trade cotton or organic cotton is one-to-one reflected in the product, and the companies and in the end the consumer can see what is the origin and is that really are these supply chains as we believe they are. So that's really just putting a barcode into materials. If we want to extend that, you might think, well, a barcode is really boring, just a number. Then we need let's say, a database where we get from, let's say, our digital number to some documentation that we have on paper on a web server or somewhere. And couldn't we be smarter with that and put real information into the product? Which is what we have in biology. And biology, let's say, if we take a piece of meat, at least in Switzerland, there's a fingerprinting done at the slaughterhouse of the piece of meat. And there's a digital backlog from the piece of meat that you have in your plate together throughout the whole life of the cow based on the DNA signature. So for the meat to the cow, DNA signature, after that, digital databases of the life of the cow. So in nature that exists. Products contain information where they come from, real information. And in digital products, we don't see that. And that's what we want to do. We want to put real information into a product, not just a barcode. And as you probably can see, with that, we're kind of combining these two things. We're combining our barcoding expertise, that we put particles and DNA into products, and we use our expertise in encoding information into DNA. And with that, we can create what we call DNA of things, where, we really put data into products. One application we've done with that is the 3D printed bunny. This 3D printed bunny has its file from which it was printed within the polymer. So you can take a piece of the bunny, clip up a small piece of the ear, take out the DNA, sequence it, decode it, feed it again through a 3D printer, and you get back the file you need to print copies of that bunny. And with that, you can make many copies of that bunny, and that's we can do that for many generations. And you could theoretically, from the digital information, create billions of bunnies out of that by really putting information as DNA into the bunny. That's similar in biology. There's a non-sexual transfer of information in some animals. Of course, the bunny can't self-replicate. It still needs us and the sequencer and the 3D printer to replicate itself. I just have to check for timing. Okay, then I'll hurry up because I thought I have until past, but I'll be fast. We can also use that technology to put information into everyday objects, also transparent objects, such as these glasses. these glasses in one of two eyeglasses contain a movie stored in DNA. What I think is really unique is that it's a transparent data storage tool, where otherwise all data storage tools we know are opaque. And here you don't see that it's there. We use this, just a few examples, in printing inks. So together with the Süddeutsche Zeitung in Germany, about a year ago, we printed a full one-day issue of the newspaper with DNA in the ink. The DNA in the ink encodes the German Constitution. It was to celebrate 75 years of the German Constitution. And every issue of this newspaper delivered to a household in its ink contains the German Constitution in DNA. And what this really shows, and my examples that will follow show, you can only do that with DNA. I don't know exactly why you would do it, but I'm sure you can only do that with. There's no other storage technology where you can distribute information in this invisible, highly compact way as here. We've done an album, we stored an album in DNA and make graffiti ink out of it, so that the artist can kind of paint with his music, because in the ink, as DNA, is the music is stored in that. And as mentioned at the beginning, we stored a movie or an episode of a Netflix series in DNA and made these nice pink capsules which were used to advertise the movie. In this case, that's 65 megabytes stored in DNA, and one of these vials contains 1 million copies of that movie, which again showcases, I mean, most of it is just water. In there are a few particles. In the particles there's DNA, and because DNA is so compact as a storage format, there's 1 million copies of that movie in that liquid. Very quickly, because I'm really fascinated for that, and sorry for going a little over time. We're really interested in randomness that we can achieve with DNA. Because as I said, generating DNA sequences with a defined sequence is very expensive, unless we just do single sequences. Or 10 or 100 or 1,000, we can do that. But if we want to do million sequences, it gets really expensive. And in physics, there's this idea of a physical unclonable function, which I very quickly have to introduce. Is you have a piece of glass, tinted glass, and if you shine a laser through it, it generates a speckle pattern. And that speckle pattern is unique to the piece of glass. And you can't recreate it, because you don't know what in that tinted glass, what is responsible for generating exactly that speckle pattern. So there's a random manufacturing process of the piece of glass, which generates a unique pattern, which enables us, or this group, to generate a physical one-way function. So if an input, a laser, into a piece of glass, and it generates a speckle pattern. Every different piece of glass generates a different speckle pattern, but you can't say in advance what is the speckle pattern a piece of glass would create. And you also can't go backwards. If you have the speckle pattern, you don't know what the glass is, and you also don't know what the position of your laser is. So it's a one-way function. You have an input, and you get a very unique output every time you do it. And it relies on randomness. And in DNA, we are really good at doing randomness, because if you do DNA synthesis and you mix all 4 bases at the same time, you get random DNA. You just get, as a chemist, this is very clear, but if you talk to computer scientists who think about randomness in a very different way as we do, this is real randomness. This is Boltzmann distribution, entropic randomness. If we do a mixture, we get random DNA. And in a, let's say, typical synthesis of DNA, we get 10 to the 16 different strands, which all have a difference. We don't know the sequence, but they all have a different sequence. We then went and sequenced a few of them to investigate the randomness of our DNA. So DNA is really good at generating randomness. But then the question is, what can we do with randomness? What can we do with, why is that unique, what we can do with random pools? And that's where these chemical one-way functions come up. And it comes back to what I introduced at the beginning, PCR. If you have such a random pool, we don't know what the composition is. It's billions of different sequences. But we know how PCR works. we know that if we take a primer pair, it will bind somewhere in that sequence to some sequences and amplify 1, perhaps 2, perhaps 10 sequences out of the pool. We don't know which ones, but we know every time we take the same primers, it'll amplify the same sequences. And so it'll amplify always the green sequence in this case, and we then sequence that and generate a binary output. So we have a binary input, which is the digital information of the primer, and we have a binary output, which is the sequences that that primer amplifies. And that response is unique to the input and the random pool. If I take a different random pool with different billions of sequences in it, a different sequence will be amplified. I don't know in advance which one, but a sequence will be amplified out of that pool. If I put in no primers, that's what the sequencing response looks like. If I take challenge A, so A primer, I do it twice, I get kind of the same sequencing output. If I take a different primer, I do it twice, a different primer output appears. I can do that for many different primer pairs and start to investigate how sensitive this is. If I, let's say, only do phase small changes in the primers, that it still give a different output, and indeed it does. And we can really relate this input to an output using randomness as, let's say, a source of identification of a pool. I'll skip this, and we've also done that in products. So we take these random pools as an authentication to put it into a product, such as a pharmaceutical product, and nobody knows what's the pool in there. It's much too large to be sequenced. It's gigantic, or it would be extremely expensive to be sequenced. But I can take pool, let's say, sample of batch 1 and let's say, and other PCR primer, and if I have the same material somewhere else, the same PCR primer, I will get the same signature from the pool. And that allows, let's say, cryptographic authentication of that, that product. You can see the small particles here in paracetamol, yeah, as the carrier of the DNA. Sorry again for being a little long. I hope I've showed you DNA is really unique. We're often asked why DNA, but I think we have extremely good arguments that this is by far the best molecule to intersect digital world and a chemical world. We can stabilize DNA in various ways, mainly by protecting it from water, also oxygen, also light. There are also reactants with DNA, but water is really important. We can use that DNA to either carry information or entropy, randomness, in a, let's say, very digital way. And we believe that's very useful in authentifying objects, putting information objects, and also as a source of randomness and entropy, where otherwise our digital world, computers have such a hard time of generating real randomness. As chemists, we're really good at generating chaos and randomness. I would like to acknowledge my collaborators and various funding sources. Thanks very much.
Thank you very much, Robert. We have time for maybe 1 or 2 questions, then there will be a break, so you could follow on with more questions to Robert. Are there any questions? Albert?
I love your work. For the PCR, multiplex PCR, you mentioned it was generally in the primer regions. Do you think it's impacts primer binding or the extension?
Yeah, exactly. We understand it affects primer binding. We also have quite a lot of data in investigating that, because really this, let's say, small loop that forms entropically really at the end, it can only form at the end because intermolecular, it's, let's say, more favorable to form at the end, and that really prevents the binding of the primer at the priming site and then the extension of the DNA, indeed, yes.
A question, yes.
Thank you, really great talk. Also a question about the PCR bias. So you mentioned that there are probably motifs that impede the amplification. Do you think the opposite is true and some motifs are actually favoring amplification and could compete with one another?
We haven't seen that. We only see, so we look at really the distribution of amplification of all of the sequences. We see that most of them amplify to the same extent. So with, let's say, some noise to the left and right. But there's like a group of sequences which really amplifies worse than everything else. So we haven't found any, but this is a, let's say, fairly large and long problem, I think, if you want to go further, because we already see in the few data sets we look at, it depends a lot on the enzyme, on the primer sets. With some enzymes, we see some other artifacts. And this is something we really have interest in understanding that better, because PCR is something we otherwise see as something really, we know everything about PCR. And also in the end, I think it's not super surprising what we found out. Let's say that there is this, let's say, binding at the tail. Still, it's completely ignored. Also, these effects of these really short sequences is somewhat unexpected, but yeah, I think there's still a lot of way we can go there, but we've never seen a positive. We haven't learned, or also the data we have doesn't show into that at all. Yes, very good question. Yeah.